Welcome to your lesson on the mitochondria and the oxidation of pyruvate. Oxidation of pyruvate is the second stage of this four-stage process that we've got for cellular respiration, as long as oxygen is available. So mitochondria, it's going to be important for you guys to know the parts and the places because you need to be able to say where each of these different stages are happening. So for example, we've just discussed glycolysis. You need to know that glycolysis does not happen in the mitochondria at all. It happens outside in the cytoplasm. So keep that locked away as well, right? Neat thing about them is that they do have their own DNA. It's circular DNA, just like the chloroplast and just like bacteria. Bacteria also have circular DNA. And so we label it for short as mtDNA, mitochondrial DNA. Then they also have their own RNA then, because it's going to read or transcribe the DNA for us. And then their own ribosomes in order to actually make the protein products. And they have 37 different genes, and they can make 13 different protein products. We wrote that on another sheet somewhere else, right? They have two membranes, two double-layered membranes. So each membrane is its own double membrane of phospholipids, just like the cell membrane. So you would imagine there'd be heads sticking out, tails sticking in on both edges of the outer membrane and on both edges of the inner membrane as well. Okay, so double layer of phospholipids, and we call the outer one the outer membrane. That's nice and easy. The inner membrane, though, it is called the inner membrane, but because it actually folds as well, you can see all these folds in here. Because it's also folding all over the place, it has another name. It's called the crista. If you're just talking about one fold, you can see that here. So it's crista or cristae for plural. Okay, so just keep that in mind. It is the same word, right? It's that whole Latin Greek pluralization thing that's going on there. So folded because what I want to do is get as much surface area as possible so that I can have tons of proteins and cytochromes, just like the chloroplast had, where you had all these machines kind of all laid out nicely to pass electrons through. I want as many of those groups as possible so that I can produce as much ATP as possible. So I can take advantage of as much space as possible to get all these machines in, okay? It is the site of oxidative cellular respiration. Oxidative in this term is referring to the redox reactions, so the bouncing of electrons between these big cytochromes and protein machines. Okay? And that's what we call the electron transport chain, the ETC. Aside from the two membranes, so the outer membrane and the inner membrane, that's also called the crista, you also have jelly on the inside here. And this area here we call the matrix. That's right, you thought it was just in a movie, but it's real. Okay, this is the matrix. The matrix is the jelly where we're going to have some chemical reactions and all sorts of stuff going on in there. So you want to pay attention to the matrix as an important location. You want to know what happens in the matrix. Okay, and then we also have, it's going to be important to us this time, in between, so in this space here, in between the inner and the outer membranes, we've got the intermembrane space. Lots of fluid in there, but you're going to see it's going to be very important because we're going to be packing our proton gradient into that space. So we pump protons into there, and then we allow them to flow back into the matrix, into the inside of the inside. And that's how we're going to use our ATP synthase. A little bit different than what we did with the photosynthetic version of it, because in photosynthesis, all of our protons were pumped into that little thylakoid disc, and then they were flowing back out into the stroma. But now we're doing the reverse. Instead of trying to pack them in and let them flow out, we're actually packing them out and letting them flow back into the center. That's the only difference between how it's going to work. Other than that, it's an electron transport chain. It's going to activate proton pumps, and we're still going to produce a proton gradient. Chemiosmosis will turn ATP synthase. Done. Okay, all that stuff will be the same. So that's your structure of the mitochondria in a little bit of a nutshell. Places are. So outer membrane, inner membrane. In between those two, there's the intermembrane space, and the inside of the inside is the matrix. So now let's do our short discussion on oxidation of pyruvate. It's very simple. This will only happen if there's oxygen available, so it is an aerobic process. And what will end up happening is you have to transport those two pyruvate molecules that I just made in glycolysis, and they have to get transported all the way into the matrix. So this doesn't happen in the intermembrane space. You have to go through the outer membrane and the inner membrane to get to the inside of the inside before you can do anything with the pyruvate. Okay, keep that in mind. So pyruvate is oxidized in the matrix. And this will be my goal up here. I'm going to want to convert the pyruvate into acetyl-CoA. That'll be my next carbon molecule. And I'm going to want to produce NADH. So I'm going to steal some electrons because that's the role of NADH, right? So convert pyruvate to acetyl-CoA, produce some NADH, and you'll see how we do that in a second. So there are 3 stages, or 3 little mini steps that we have to do. First of all, if you look down below here, you can see there is this pyruvate molecule. I've already shown the red line where it chops it in half. But the bottom of it is called an acetyl group, and the top of it is called a carboxyl. And you already know that carboxyl is really, when I chop it off, it'll be a carbon dioxide, right? You can even see it's a COO. So the first thing I'm going to do is transport the whole thing. I'm going to use a protein in the inner membrane to do that. Transport the whole thing to the inside. Once it's inside, I'm going to chop off that carboxyl group, and you can see it coming out as, here's our arrow here showing it coming out as the CO2 at the bottom. That means I'm just left with this acetyl group right here. This 2-carbon molecule at the bottom. That's all that's left. I'm going to, at the same time that I'm stealing that carbon dioxide, I'm going to take advantage of that, and I'm going to convert NAD, reduce it to NADH, so steal some electrons. Okay? And then, so that I can make it useful for the next group of enzymes that are going to do their job, I have to add on something called a coenzyme. This particular one is coenzyme A, so CoA. This is going to help the acetyl group fit perfectly into the active site of the next enzyme. That's why it's called coenzyme here. It's helping us to do the next stage, okay? So we have lost, for the first time, we have actually produced a carbon dioxide. This is the one of 6 of the original carbons that came from our glucose. Keep in mind, though, that we had 2 pyruvates, right? We took the C6 sugar, the glucose, we cut it into 2 pyruvates. Each of them are C3. They each have 3 carbons there. So now I've taken one of them and I've chopped it down to a 2-carbon molecule called acetyl. I'm going to do that twice, which means that really, because this runs twice, I've actually lost 2 carbon dioxides. 2 pyruvates, 2 carbon dioxides, 2 acetyl-CoAs is really the overall picture. So this whole thing is times 2. That's it. That's the oxidation of pyruvate. I want to point out something, though. When I cut off a carbon dioxide, I stole electrons. That's important. Remember, tuck that away because it'll help you memorize the next stage. When I cut off the CO2, cut off the carboxyl group, I also stole electrons, and I can make an NADH out of that. Okay, where am I? I'm in the matrix doing this. This entire thing is in the matrix. So transport the pyruvate inside the inside first, then you start chopping off stuff and stealing electrons and shoving on the CoA. If we're dealing with what's in and what comes out, you can see we put in 2 pyruvates, eventually, like one at a time, but we put 2 pyruvates in there. That means that we needed 2 NADs, which are here, which of course means that we will make 2 NADHs on the other side. Then we got a couple CoAs we need to put in because they're going to become part of the acetyl-CoA section. And of course, we started with a 3-carbon pyruvate. We ended up with a 2-carbon acetyl. So there's our other carbon there sitting in the carbon dioxide. I mentioned to you before that you want to make sure you follow the carbons and you want to follow the energy. So here we've got circled in green the 2 pyruvates. That's a carbon molecule. You can see it becomes the 2 acetyl-CoAs, and the carbons are also becoming the carbon dioxides. That's where we're splitting things off. So you follow those as your own conversation. And then you can see we've also produced NADHs. That's our energy. We're following the electron energy as well as any ATP that gets created. So that's why I have underlined these suckers for you. Just like in our conversation when we talked about when you split the water in photolysis back in photosynthesis, we wanted to know like what happened to the oxygen, what happened to the protons, what happened to the electrons. Well, in this case here, let's talk about what happens to the parts that we've just made. So the CO2 that we've just created, as far as the mitochondria goes, it's useless. This is the lowest form of energy. In this particular process, this is the waste. So we're just going to get rid of this garbage. Now, if I'm a plant cell and my mitochondria happens to be pumping out carbon dioxide, Well, maybe that carbon dioxide makes its way to my chloroplast. But if I'm an animal cell or any other type of cell that doesn't have the chloroplast, cannot make their own food through harnessing light energy, then for sure CO2 is going to diffuse out of the cell. It's going to go into my bloodstream, and it's going to be carried to my lungs. It's going to diffuse into the lung sacs, to the alveoli, and then it's going to be breathed out by me. So this is complete waste. We have no use for carbon dioxide. Carbon dioxide actually will begin to form an acid as soon as it hits my blood, and that acid will start to interfere and denature enzymes and proteins. So I really don't want this acid sticking around. It becomes an acid, carbonic acid, when it hits water. It forms a chemical reaction. So I want to get rid of the sucker. It's going to be gone from me. The protons, well, they're pretty useful, actually. Protons are going to be used to drive proton pumps, just like we saw in photosynthesis. So they're actually going to stay in the matrix, and then when an electron passes through the membrane, I'm going to pump them out into the intermembrane space, and then they're going to flow back through and drive the ATP synthase and make me some ATP. So they're actually going to be pretty useful. Protons are all over the place. And then the NADHs that I've just created are my electron carriers. So they're going to deliver those electron payload to my electron transport chain to help me drive chemiosmosis and help these protons get moved around to help me produce ATP. This is what we call oxidative phosphorylation. Because again, it's the movement of electrons through a membrane that activates pumps. And those redox reactions, those oxidative reactions, are going to pump those protons and allow ATP synthase to work. Any ATP made that way is oxidative phosphorylation. Any ATP made by an enzyme who steals a phosphate and gives it to an ADP, well, that would be substrate level. And you really do, I'm going to say quite a few times in our lessons because I really want it to stick for you. But this idea, using electrons to drive this process, is oxidative. So what's going to happen is the electrons will move through the cristae, so that inner membrane that's folded, and they're going to activate the proton pumps. That'll give us a proton gradient in the intermembrane space, a nice high concentration, flows back in through ATP synthase, cranks the head of that ATP water wheel kind of shape there, that motor, and ADP is phosphorylated to ATP, done. Next thing we have then is our major carbon molecule that we care about, and that would be the acetyl-CoA. The acetyl-CoA, actually at this point, what I do with it depends on my body's needs. If I actually have enough ATP, and I really don't need to go and produce ATP at this time, then maybe I won't go chop up the acetyl-CoA in the next cycle, which is called the Krebs cycle. Maybe instead what I'll do is convert it into fats, as you can see written at the bottom there. So rather than going through Krebs and making ATP, maybe I'll just make fats. Because it's a 2-carbon molecule, it's pretty easy to just stack them all together in big, long fatty acid chains. Remember, those fatty acid chains are those huge, big, long carbon chains, right? So that's pretty easy to create. The other thing that's neat about acetyl-CoA is that I can pretty much make it from any type of macromolecule that I have access to. So I can chop up a protein. Remember, amino acids are NCC as its backbone, the nitrogen and the carbon-carbon. So it's got an amino, the central carbon, and it's got the carboxyl group, right? Well, why don't I just take that CC, that 2-carbon part of the amino, chop off the actual amino group, so get rid of the nitrogen, and now I've got myself an acetyl. Great. Done. Pretty easy to do that. Lipids, if I can create fats with it, then I can go the other direction, and I can take every 2 carbons and I can cut them up and make them into acetyl-CoAs and run them in this direction. Carbs. Man, I've got so many chunks of acetyls. We've just shown you how we can chop up glucose and then cut down to an acetyl, right? Chop off a carbon dioxide. So it is called the central molecule. That's the fancy name for it. For energy metabolism. Anytime you see the term metabolism in this sense, we're talking about energy production. Breaking down molecules for energy, all that kind of stuff. That's metabolism. If we have oxygen, this is what's going to happen. We will steal electrons. So that's why it's the oxidation of oxidation of pyruvate, I should say. So we're going to steal electrons, chop off the CO2, shove on this CoA so that we can move to the next stage. If, however, I don't have oxygen, then the pyruvate will do what we discussed in the fermentation lesson. Pyruvate could be converted into ethanol and a carbon dioxide, and or it could be converted into lactic acid, which thankfully is a reverse process that we can do, or a slew of other things. We've only mentioned 2 of the many things that can happen to pyruvate. All depends on the type of organism that you are and what you have access to. Okay? And again, you can go back and take a look at that if you wanted to refresh your memory on the anaerobic. So that is actually the end of our mitochondria and oxidation of pyruvate conversation. It's that simple.